In my talk, I want to present some of our fMRI work aiming to bridge between macroscopic and mesoscopic scales in the human brain by exploiting the high resolution of ultra-high field MRI scanners. In the context of the Human Brain Project, we aim to combine neuroscience, modeling, and computer simulation to gain a better understanding how processes at lower levels explain processes at higher levels. The work I present today operates at the higher levels of the depicted scales on the left side using functional brain imaging. Conventional fMRI is a useful method to study macroscopic scale functional properties of areas and networks, but provides limited constraints for modeling brain function at the level of more fine-grained representational contents. Ultra-high field fMRI at 7 Tesla and above achieves sub-millimeter resolution, thus providing the exciting possibility to resolve functional activity in mesoscopic scale structures, such as cortical columns and cortical layers. While I have not the time to present details, the presented results are used to constrain our efforts of building biologically inspired large-scale neural network models, and the data I'm presenting is also becoming part of the Human Brain Project Atlas. The first study I'm presenting does not directly attempt to resolve columns or layers, but exploits the higher signal-to-noise ratio of ultra-high field fMRI to advance our knowledge about generative top-down networks supporting mental imagery. More specifically, we wanted to investigate whether top-down effects in primary visual cortex are topographically organized and strong enough to reconstruct imagined letter shapes from the mind's eye using ultra-high field fMRI. In the first encoding step, we basically used conventional standard retinotopic mapping in visual cortex using the so-called population receptor field approach from Dumoulin and Mendel. And we used that approach to obtain a mapping from positions in the visual field to locations in visual cortex, especially in areas V1, V2, and V3. In the second decoding step, we basically invert the process by projecting the PRF locations of voxels activated during a specific imagery trial back into a visual field representation, thereby reconstructing the imagined shape. The slide shows the result first for the case of presented letter shapes where subjects see the stimuli. The reconstructions shown above the letter stimuli are based on voxel activity in primary visual cortex of a participant, showing that the decoding process works successfully since the reconstructions clearly resemble the shapes of the presented letters, here the letters H, T, S, C. Here we see in the upper row the shape reconstructions when a participant merely imagines the respective letter shapes. The results are much less clear than in case of seeing the letter shapes, but analysis shows that the reconstructions contain enough topographic information to identify which letter has been imagined. Note that the generated top-down signals reaching V1, V2, V3 are very weak and noisy, and we were even unable to decode letter shapes when using only 3 Tesla fMRI. In order to improve the reconstructions, we used multi-layered neural networks as an additional pre-processing step of the obtained brain activity patterns during imagery. More specifically, we trained a denoising auto-encoder network using only brain activity from trials of the perceived letter stimuli. To these patterns, we added noise to these activity patterns and asked the denoising autoencoder to recreate the noise-free patterns which we had obtained in the perception trials. We hoped that based on this training, the network would also denoise activity patterns obtained later during mental imagery. This slide shows in the upper row the construction results when running the imagery-generated activity patterns in early visual cortex, first through the trained denoising autoencoder and subsequently performing the PIF decoding step. As you can see, the achieved reconstruction quality has substantially improved and the letters are easily recognizable. Given these results, the currently developed system is transformed in a real-time version of letter imaging decoding in form of a brain-computer interface. And we hope to apply it to people with motor impairments which cannot speak. The next examples explicitly exploit now the achievable sub-millimeter spatial resolution of ultra-high field MRI to really enter now the mesoscopic space. The possibility of to measure activity in the cortical layers and columns opens a new window into brain function, which is not achievable with conventional 3 Tesla fMRI. With a cortical thickness of 2 to 5 millimeters, the currently achievable functional resolution between 0.5 and 1 millimeter is able to provide about 3 to 4 independently sampled voxels along cortical depths. While this resolution does not allow to resolve all cortical layers in gory detail, the resolution is good enough to separate upper, middle, and lower compartments, which is important since this provides information whether a region is primarily driven bottom-up or top-down. Besides the laminar organization, many brain areas of the primate brain are organized in cortical columns along the cortex, and the examples presented next are focusing on this columnar organization. A cortical column consists of thousands of neurons responding roughly to the same feature, for example, horizontal versus vertical orientation bars in V1 or different directions of motion in area V5. Being able to resolve such columnar feature clusters allows us to measure the coding principles inside small specialized areas, as opposed to measure the response properties of the mean activity of a whole cortical area. Analyzing cortical layers and columns requires extremely high quality segmentation of gray matter, with a precise demarcation of both the inner white gray matter boundary as well as the outer gray matter CSF boundary. To achieve the required quality, usually required substantial manual editing work to create good segmentations, and in our case of sub-millimeter resolution, it was also necessary to segment small blood vessels, to mask them out because they distort the local fMRI signal. In order to improve automatic segmentation for sub-millimeter ultra-high field anatomical data, we developed a state-of-the-art semantic segmentation network called Tiramisu. While I have not the time to present the network in detail, the network is highly powerful for our task since it requires only a few data sets to train and provides excellent generalization results. Here's an example applying the Tiramisu net to a new 70 dataset. As can be qualitatively seen, the segmentation is indeed of unprecedented quality. Another qualitative comparison to conventional segmentation tools shows that the results are indeed outperforming traditional segmentation tools. Here we see, for example, segmentation errors in the front and back on the left side on the FSL fast version, which are not made by the UNet, and also network we trained and tested, and on the right side by the Tiramisu net. A more quantitative comparison shows the increased segmentation accuracy of the tiramisu net in comparison to 3 conventional neuroimaging segmentation tools. Here, the dice coefficient measures the overlap between the obtained segmentation with the ground truth expert labeling of held out validation datasets. Also, another commonly used metric, the Hausdorff distance, reveals the superior segmentation results of the tiramisu network. While the obtained high-quality gray matter segmentation is sufficient, we can now model the cortex as a 3D structure with 2 dimensions running along the cortex and 1 dimension running along the depths of the cortex. To achieve this, we model the cortical depths in this kind of 3-dimensional space by reconstructing inside gray matter so-called regular depth grids along any desired depth level. These regular grids are connected through depths at corresponding grid intersection points to model the local orientation of cortical columns. Our modeling technique also models the curvature-dependent change in position of layers, which is commonly related to the so-called equivolume model to correct for this shift. The reconstructed 2D regular depth grids are shown here on the left side, and from those we model layers filled by voxels as shown on the right side. And these structures can then be used to sample functional data and to assign functional data, which is aligned to the anatomy, to the various depth levels and columnar structures. Our regular depth grid modeling technique also allows us to visualize smaller cortical regions as flattened 3D volumes, which allow to apply many tools developed for volumetric data. The picture here shows 2 measured features in green and red, and we can see that they largely form columnar clusters extending through the depicted depth dimension. I'm now showing an example application of this technique, which was the first application of columnar mapping in extrastriate cortex. Using conventional fMRI, motion-selective area V5 IhMTAVS has been routinely mapped in many labs by contrasting moving stimuli to static stimuli, as shown in this slide. Using 7 Tesla fMRI and the described cortex modeling technique, we asked whether we can map the features in this area, such as the direction of motion columns, described by Newsom and colleagues in the non-human primate. The slide shows for one participant that we could indeed reveal different axes of motion columns in area V5. The upper row shows the modeled extent of depths in area V5, while the lower row shows the columnar organization obtained when presenting stimuli moving back and forth in different directions. The colors thus correspond to so-called axes of motion columns, corresponding to e.g. horizontal motion or vertical motion or oblique motion direction pairs. This slide also shows in the top row that responses were rather consistent across depths, but also exhibited substantial variability. The lower row shows that calculated cross-validated tuning curves obtained for different axes of motion columns were very similar to what we expected from electric recordings in macaque monkeys. In the final example of mesoscopic imaging, I want to present recent work where we aimed to relate the contents of conscious percepts to the fine-grained level of motion direction specific cortical columns. This slide shows an example of apparent motion. You might see a white square jumping to the right and left, and this kind of motion is induced by the brain because here only pacments open and close at the left and the right side, but there is no physical motion in that scene. So our brain constructs the percept of motion, which is called apparent motion. When we analyzed that data, we found that area V5 HMT+ is the first region which strongly responds to the motion, induced motion versus flickering control conditions. So we reasoned, and this is confirmed in many labs, that area V5 is kind of the source of constructing the percept of apparent motion. In this high field application of apparent motion, we first mapped, similarly as shown before, the horizontal and vertical axis of motion, now only these two opposing directions of motion, as a first step for a ultra-high field experiment. This is the main stimulus. This is the apparent motion stimulus. They are simply shown two squares, either oblique left upper right lower corner or left lower right upper corner. And this is an ambiguous stimulus. So you can either see the stimulus moving horizontal or moving vertical. This is so-called motion quartet stimulus. And we now wanted to measure and relate whether the conscious percept of horizontal versus vertical motion was related to the axis of motion columns in area MT. We found and mapped these two axes of motion in area MT as shown here on the left upper side, and the activity during physical motion of horizontal and vertical motion versus the induced ambiguous horizontal vertical motion were largely overlapping as shown on the left. The right side shows that also our clusters usually showed quite nice vertical consistency in depths. So we kind of revealed, again, columnar organization within MT for these two opposing motion directions. More importantly, the motion effects indicated then when subjects perceive, indicated by button press, that they now perceive the ambiguous stimulus as horizontally moving, the vertical columns in MT ceased activity, while the horizontal clusters increased activity, and vice versa when the percept switched from horizontal to vertical motion. To show this in a bit more simplified way, this movie highlights these results. You see here voxels in HMT, and the percept switches now to horizontal, and the red voxels increase, which correspond to horizontal columns. And now when the vertical percept dominates, the vertical clusters increase. Now again, the red for horizontal perceived motion, now again, the green for vertical motion. This is speed up, so the switches are in the order of 5 to 10 seconds, but this is from real data of one participant. This concludes this short voyage into the possibilities of mesoscopic fMRI with a focus on columnar feature mapping. I would like to note that the presented datasets are curated and made available in the Human Brain Project's eBrains digital infrastructure, and that some datasets are also soon available as part of the HBP Brain Atlas. Finally, I want to thank my collaborators and funding sources, and especially you for your attention.